Hello friends, I am back with my last video of chapter 3 Inheritance and Variations. Now we would start with linkage. As we know, many genes are present on chromosomes, and if two or more genes are inherited together are known as linked gene, and the tendency of transmission of those genes is known as linkage. The linkage was first discovered in plants by Betson and Punnett, and in animals it was discovered by T.H. Morgan. Linkage is always of two types. One is complete linkage and second one is incomplete linkage. The complete linkage, the linked gene which are closely located on chromosomes and do not separate are known as completely or strongly linked genes. Examples: X chromosomes of Drosophila males. Second one is incomplete linkage, the linked gene which are distinctly located on same chromosomes and have chances of separation by crossing over is known as weakly or completely linked gene. Example in GMA's color, shape and of the grains. Then sex linkage, the transmission of X-linked and Y-linked genes from parents to their offspring is known as sex linkage. The sex-linked inheritance is of three types: X-linked, Y-linked and XY-linked inheritance. And the crossing over. The crossing over is the process that produces new combinations of genes by interchanging and exchanging of corresponding segments between non-sister chromatids of homologous chromosomes. It occurs during packaging of prophase 1 of meiosis, and it consists of four sequential steps: synapsis, tetrad formation, crossing over and terminalization, which we had already studied in 11th standard chapter named Cell Division. Now we will study about Morgan's experiment on Drosophila. So Morgan used Drosophila melanogaster, that is also commonly known as fruit fly, for his experiment because there are few reasons behind that. One was that Drosophila can easily be cultured in lab. Second one, its lifespan is very short, that is about 2 weeks only. And third one, it is having high rate of reproduction. So it was used by Morgan's experiment for checking the dihybrid cross in fruit fly to study the genes that are sex linked. Here is Morgan experiment showing linkage and crossing over in Drosophila. Here you can see two crosses are available, one cross and cross 2. In cross 1 you can see that the male is having Y and W genes which are closely located on the chromosomes, yellow and white, that is orange colored, and the male which is Y plus and W plus, they are also closely located genes present on the chromosome, and that is of wild type. When they are crossed with each other in F1 generation, hybrids are formed where wild type and yellow white types are formed, and in F2 generation you can see that the parental type is 98.7% and recombinant type is 1.3%. So by this cross you can see or you can identify that if genes are closely located on the chromosome, then the parental type is maximum than the recombinant types. And in F2 generation, the recombination happens or the crossing over happens between Y and W genes present on the chromosomes. While in cross 2, you can see that the male is wild type again, which is denoted by W plus and M plus, and the female which is denoted by W and M, which is white and miniaturized. So they are crossed, and but you can see here that the genes are distinctly located with each other on the chromosomes. That is why in F1 generation obviously hybrid will form, but in F2 generation then the parental type is 62.8% while recombinant type is 37.2%. So you can see when the genes are located distinctly located on the chromosome, then the recombinant type is more than when the genes are closely located. Also the recombination or crossing over happens between W and M genes present on the chromosomes. Here The genes for yellow-bodied and white-eyed were strongly linked and showed only 1.3%, which we had already seen in the picture. The white-bodied and miniature wings showed 37.2% recombinant gene in cross 2. Cross 1 shows crossing over between genes Y and W, and cross 2 shows crossing over between the genes white and miniature wings, and the dominant white type alleles are represented by plus sign. Then our next topic is autosomal inheritance, the transmission of body character other than the sex-linked traits from the parents to their offsprings through autosomes is called autosomal inheritance. Some characters which are influenced by dominant genes, while some are by recessive genes present on the autosomes. Example, autosomal dominant traits examples are widow's peak, Huntington's disease, and autosomal recessive traits phenylketonuria, cystic fibrosis, and sickle cell anemia. These are the example of autosomal dominant traits, and these are the example of autosomal recessive traits. Now, what is sex-linked inheritance? The inheritance of sex-linked gene located on non-homologous region from parents to their offsprings is called sex-linked inheritance. X-linked inheritance examples are hemophilia, color blindness, night blindness, myopia, and muscular dystrophy. And Y-linked traits are inherited directly from male to male. Example is here on ear pinna. Now, sex determination in human being. The chromosomal mechanism of sex determination in humans is XX and XY type. In human being, the nucleus of each somatic cell contains 46 chromosomes or 23 pairs of chromosomes. 22 pairs are autosomes and 1 pair is known as sex chromosome. Thus, the genotype of female is 44 autosomes plus X and X, and in case of male it is 44 autosomes plus X and Y. So here is the sex determination in human being. Here you can see in parent generation one is male and one is female. X and Y is denoted by male, X and X is denoted by female, and the gamete will formation XY and XX. Then the fertilization will happen. F1 generation, the hybrid will form. So you can say here in this checkerboard, you can say first is female, XX cross will become a female. In X and Y will cross, it will become a male. When X and X will cross, then it will become a female, and then X and Y will cross, it will become a male. So your conclusion is that the ratio of sex producing in any generation will be 1:1 only. So that indicates that the sex of a child always depends on the type of sperm fertilizing the egg, and hence the father is always responsible for the determination of sex of a child, not the mother. So it is always known that the mother is responsible for determination of a sex, but here by the diagram you can see that the father or the sperm always responsible for the determination of sex of a child. Next, you can also read sex determination in birds and honeybees also. I am not going to explain here in details of birds and honeybee. You just can study through your textbook. Next, we will see the genetic disorders. Two categories are there. One is Mendelian disorder, caused mainly due to the alteration or mutation in the gene. The example, thalassemia, sickle cell anemia, color blindness, hemophilia, and phenylketonuria. And second one is chromosomal disorder, that are caused due to the absence or excess of one or more chromosomes of their abnormal arrangements. Example, Down syndrome, Turner syndrome, and Klinefelter syndromes. These genetic disorders are really important from your examination purpose. So let's take one by one. First one is thalassemia. Thalassemia is an inherited blood disorder wherein the body produces an inadequate amount of hemoglobin. It develops when there is some abnormality in any one of the genes that are involved in the production of hemoglobin, and this defect is inherited from the parents. There are two different types of thalassemia, alpha thalassemia and beta thalassemia. Alpha thalassemia, a disorder in which one of the gene of alpha globin has a mutation or abnormality. Beta thalassemia, the genes of beta globin are infected. So that is about thalassemia. The next is Down syndrome. Down syndrome is a genetic disorder caused when abnormal cell division results in an extra copy of chromosome number 21st. Always remember that the 21st chromosome is having an extra copy of chromosome. So this extra genetic material causes the developmental changes and physical features of Down syndromes. These individuals will have 47 chromosomes instead of 46, that is normal number. So 46 is a normal number, but in case of Down syndrome, the child will always have 47 chromosomes. So here this diagram you can see that the 21 number chromosome is having 3 chromosomes rather than all the chromosomes they are diploid, but here this 21 number is always having 3 chromosomes. So that is how this is denoted, the Down syndrome, which is known as trisomy 21. The next is Turner syndrome. Turner syndrome is also known as X monosomy or XO females. So Turner syndrome has 44 autosomes with XO. Okay. So Turner syndrome is a condition that affects only females, results when one of the X chromosome is missing. X chromosome, we are talking about sex chromosome. When X chromosome is missing or partially missing, the symptom of Turner syndromes are short stature, non-functioning ovaries, webbed neck, puffiness or swelling of the hands and feet, and skeleton abnormalities, heart defects, high blood pressure and kidney problem. So here you can say that the Turner syndrome is only related to females where sex chromosome is missing or partially missing. In this diagram you can see this last square, you can see 2 chromosomes are there, but then only one purple chromosome, which is X chromosome, you can see. Second chromosome is little colorless, which is loss of second X chromosome, that is partially loss of that chromosome, that cause monosomy or Turner syndrome. The next one is Klinefelter syndrome. Klinefelter syndrome is a genetic condition that results when a boy is born with an extra copy of X chromosome. Klinefelter syndrome is a genetic condition affecting males and it often isn't diagnosed until adulthood. So this is a characteristic which is not diagnosed until adulthood. The genotype of the individual is X44 plus X, X and Y. The symptoms: harsh voice pitch, underdeveloped testes, feminine development and no spermatogenesis, therefore individual will be sterile. So there is a structure or a diagrammatic presentation of Klinefelter syndrome. In the last circle you can see there are 3 chromosomes. Instead of 2 chromosomes, there are 3 chromosomes. So 2 XX, which are large in size or longer in size, and 1 Y chromosome is also present, which is smaller in size. So if the combination of those chromosomes is available, that person is having Klinefelter syndrome, which is mostly related to the males only. So this is how we are ending this chapter, Inheritance and Variation. Hope the videos will help you in understanding that chapter properly. Thank you for watching.